Well, it is just about 8 o'clock on the dot. Thanks everyone for joining us today for our final virtual Clean Lakes 101 Science Cafe. Very excited. Next month we'll be over at the Edgewater for a little more fun. All right, well, let's get things started. My name is meteorologist Max Paris. I do the weather here at Channel 27, the ABC station in southern Wisconsin. We're proud media sponsors working with Clean Lakes Alliance. And, you know, we're entering into our spring months now. It still feels wintry out there, but of course, when we think spring, we think Easter, we think about Lent for religious holidays. And this kind of works with that. We're starting to think of our Fish Fry Fridays. And the title of this morning's 101 Science Cafe talk is called Microbes and the Mercury in My Fish Fry. I heard this topic and I'm like, uh-oh, what am I eating? So microorganisms are responsible for the high levels of mercury, which is a toxin frequently found at unhealthy levels in our fish, including some of it in our Wisconsin lakes. So those microbes that cause the mercury have extraordinarily diverse physiologies, communication strategies between them, and the mechanisms of their evolution. So today we will learn how microbes work with mercury, and discuss the consequences for biomagnification of toxic mercury in aquatic food chains. So it's an interesting sort of stepping stone and then eventually ends up on our plate. So here to introduce today's speaker is Heather Vogner from First Weber. Morning, Heather.
Good morning, everyone. Thanks, Max. Along with Johnson Financial Group, First Weber is proud to be presenting sponsor of these talks. Joining us supporting Clean Lakes 101 is our hosting sponsor, the Edgewater, and supporting sponsor, National Guardian Life Insurance. Our production partners are the UW-Nielsen Institute for Environmental Studies and the UW Extension. And as Max has said, we are happy to have WKOW as a media sponsor. Two quick things before we get started. First, Clean Lakes annual campaign is underway. If you haven't renewed your individual friend donation or business or organization lake partner donation, now is the time. You can visit cleanlakesalliance.org to make that donation. Remember, these talks are free all year long to friend and lake partner donors. Second, registration is open for the annual Loop the Lake bike ride. Ride in person on Saturday, July 18th, or virtually the week leading up to the 18th. Registration info is on the screen. All riders receive a super soft t-shirt, compliments of Land's End, and as always, kids 10 and under ride free with a paid adult. Back to today's talk, which, as Max has already titled, Microbes and the Mercury in My Fish Fry. Here to present today's topic is Dr. Trina McMahon, a professor of civil and environmental engineering and bacteriology at the University of Wisconsin-Madison. Dr. McMahon's research interests include microbial ecology, water quality, and environmentally biotechnology. She has authored more than 70 research articles with her graduate students and is passionate about training future faculty. Please welcome Dr. Trina McMahon to tell us a little bit more about this very interesting topic.
Thanks so much, Heather and Max. I should be able to share. Let's see, of course, now you're probably seeing my family. Let me try this one. All right. Somebody want to give me a thumbs up or?
Looks good, Trina.
Looks good. All right, great. I will turn on my presentation then. So thanks everybody for showing up on Zoom. I'm sorry that we can't be in person. I think the last time that I spoke at a Clean Lakes Alliance event, it was at the Edgewater and must have been pre-pandemic. I talked about cyanobacteria in Lake Mendota. And so I thought I'd go for something different today and talk about mercury. This is a relatively new area of research in my lab, maybe for the past 4 years or so. And it's been really fun to learn about something that I hadn't really thought about much before. I eat a lot of fish fry. I didn't before I moved to Madison about 20 years ago, but I think it's been just about every Friday since I moved here that I've been in town that I get my fish fry. In fact, my son was born on a Friday night after a fish fry. And yeah, you'd think that I wouldn't eat it anymore after that but no, it's all good. So I'm going to tell you today about just a little bit about probably the stuff that matters the most to you, which is how much is in the fish. And then I'm going to tell you more about where the mercury comes from and the role that the microorganisms in the lake play, because that's my focus. I study microbes. I'm both a microbiologist and a water quality engineer, which is an odd combination, I know, but someday I'll explain it. And let me make sure I can advance. Okay, so we already talked about the importance of fish fry, especially during Lent in Wisconsin, but it's just a really ingrained part of our culture. This is, going to make you nice and hungry sitting here. I did look up the history of fish fry just a little bit in preparation for this, and it is, of course, tied to Lent, but it became such a tradition to go out for fish on Fridays, even before it was just by the church considered to be only a Lent thing. It was an every Friday thing. And so people just, even after the recommendation was to only do it during Lent, people were so accustomed to going out and getting their Friday fish fry that it was just a really central part of our Wisconsin culture. Maybe you've noticed that you can get different kinds of fish. You can get walleye. That's my favorite. A lot of places have cod, which, wouldn't be from a freshwater lake, but there's perch and bluegill and, all the things that you could fry up that you have ready access to, people will eat as their fish fry. Now, maybe you've also heard that there are fish consumption advisories. It's not just with fish fry, of course, it's with any fish that tends to be high in the food chain, meaning that it is long living and eats smaller fish, which is the case for most fish. But if they eat medium-sized fish and those fish eat smaller fish, then we get what we call biomagnification, which I'll talk more about in a minute. Most of the fish that we eat, but not all, in fish fry regularly are pretty higher up in that food chain. So what I'm showing here is a graph in the top right that shows data from the Wisconsin DNR for an average period of 1970 to 2012 for 3 lakes in the Yahara region. And you can find your favorite one on there if you want. And I guess the takeaway is that these boxes are a condensation of data that shows sort of the average, which is this dark line in the box, and then the range. So some fish will have a lot and some fish will have not as much. And the lines that go horizontally show the ranges between which the consumption of that fish is considered to be, I don't want to say dangerous, but not advisable for women of childbearing age and children. So that's the dotted line. For consumption once per month. Now the solid line is for consumption once per week. So that means, or it's the other way around, I'm sorry. So once per month is the solid line. So you shouldn't eat them as often because they have more mercury, right? So that's how to remember the relationship there. So if a fish has less mercury, like in this region between the dotted lines, then you can eat it more often, like once a week. And this is based on estimates of toxicity of mercury to mainly to developing fetuses, because it can cause developmental defects. It's a pretty potent neurotoxin, and we worry about it the most in women of childbearing age and children. There's less restrictive advisories for men and children over 15. But there's still enough in Lake Monona in particular that there is this advisory showing that you really should only eat walleye from Lake Monona once a month. So if I cooked my own fish fry, that would cut into my weekly consumption. Now, I think one of the more disturbing things about mercury in our Yahara lakes is the fact that there's a lot of there's a portion of our community, our human community, that relies a lot on the lakes for their protein, right? That they're out there fishing on the ice, like the picture that Max showed, and they eat a lot of fish per week, per month. And this chart shows on the left how much fish is being consumed by these different populations in our community, and showing that it's also different, particularly between Hmong American and African American. They eat different, it's in their cultures to eat different kinds of fish. And so this study that I'm citing right here was exploring whether the differences in fish consumption preferences would relate at all to their mercury intake. And what they found was that because Hmong Americans in particular consume, what ends up being this gray bar here, which is the white bass, they get exposed to more mercury because that fish has more mercury in it. And so this study was actually done by a group here at University of Wisconsin-Madison. The person that led the study, Pete McIntyre, is now at Cornell University, but it's a really interesting study, and I believe that it's open access. So if you wanted to go and take a look, it's super interesting, and it's very readable. Like, it's not, I mean, it's a scientific paper, but I found in an area that's not what I do. Actually, I don't go and survey human populations for what they eat. It was very easy to understand. So it's very worth checking out. And yes, so the red text means that those fish aren't, they recommend for women of childbearing age and children to not eat more than 1 meal per month. And the fish consumption rate per day, to get a sense of what I'm eating every week, I eat about 250 grams at my weekly fish fry. And so to put this in perspective, this is how much these populations are eating per day. So this makes us worry about the mercury in the fish in Lake Monona and Mendota. Mendota doesn't have a fish advisory, but there's been some research done, not by the DNR, but by the UW, on the amount of mercury in Lake Mendota. And it is still enough to be worried about the fish that are in Lake Mendota. I think the sources of mercury in the two lakes is different, and I'll talk just a little bit about that. And so it's possible that the fish in Monona are bioaccumulating more of the mercury because of where the mercury is and what its source is in Lake Monona versus Lake Mendota. So this is a fairly busy graph, or image, but it shows kind of what we call the mercury cycle. And it's, I have a more complicated one coming up, but this one focuses on the sources. So mercury is naturally occurring in volcano eruptions, and it's also just in ore, that when people mine certain kinds of metals, particularly, there's mercury in it, in that ore also. And so, like, one of the most common sources of mercury poisoning in the world is artisanal mines. So especially in Africa and South America, where there's a lot of people that have their own boutique small mines. And so the people working there get exposed to tremendous amounts of mercury. We don't have that so much in the United States. Our sources are largely coming from coal-fired power plants. And maybe you're familiar with acid rain and how the sulfur that's in our coal, when we burn it, goes into the atmosphere and deposits on the landscape. Well, the mercury co-occurs, or is in the same coal as the sulfur. So where we have problems with acid rain, we often also have problems with mercury contamination. And it's global, because the mercury can travel through the atmosphere at very high up in the atmosphere, and then eventually come out in the rain or the snow. And so there's mercury all over the planet. There's a lot of people studying it in the Arctic, even though it's far away from coal-fired power plants. There's quite a bit of bioaccumulation that happens in Arctic animals. Closer to home, we think that the mercury in Lake Mendota and Monona are, like I mentioned, coming from different sources. We think there was an industrial source historically in Lake Monona. Lake Monona also gets a lot more urban runoff than Mendota, and obviously Wingrid does too. And so we think that the mercury in Lake Mendota is coming more from this atmospheric deposition that may be concentrated by the Yahar River coming in and delivering what was on the landscape north of the lake, kind of like phosphorus. But Monona has more of this reservoir of mercury that maybe historically was discharged there from some industrial activity. And there's actually a research group at UW studying exactly that. If you ever come across Grace Armstrong, she's a PhD student working with Jim Hurley on that exact question. So we collaborate a lot with her and with Jim's group. They focus more on the higher parts of the food web. We focus on the microbes. So you can see that the arrows show like where the mercury is coming from. And what I'm most interested in is the part in the lower left, which in this graph just says bacterial conversion to methylmercury. And then it shows that the fish, as you go from small fish to big fish, can get mercury accumulating in the food web. And so I'm interested mainly in what's happening down here. And this is a more complicated image, so I'm taking it up a notch here. There's lots of chemistry going on. So if you don't want to remember your chemistry, that's fine. I'll point out the most important things. Kind of boggles my brain when I look at it, actually. So it's also showing the source of mercury that can come from the atmosphere. This shows oceans, but like same thing happening on the land and in lakes. The most important thing to notice here is that what we care about is how much ends up in the fish, but that comes from the very base levels of the food web, like the bacteria and the phytoplankton, which would include algae and cyanobacteria, but then also zooplankton and then small fish, and all the way up to our walleye and muskies. Now, the form of mercury that we worry about the most is right here. So we call it MEHG is what you're going to see in the rest of my presentation. So methylmercury, and methylmercury is what I would call we call it the organic form of mercury. So the inorganic form, which is what is in the atmosphere, and what is shown here, anytime you see an Hg0 or an Hg2, that means that there's no carbon atoms on it. And when there's no carbon atoms on it, it can't really accumulate in an organism. And so if the mercury in the system is staying as inorganic mercury, it's not going to hurt much of anything because it can't get into the organism and stay in the organism. So what we worry about is when it becomes, when it turns into an organic form. And I'm not talking about organic at the farmer's market. I mean, like chemically, in the sense of having carbons and oxygens and hydrogens on it. It's the definition of organic. And what you'll notice is that down here, this SRB, these are very specific kinds of bacteria that need a very specific habitat in order to live and make a living. And they are the ones that are responsible for producing the methylmercury. That is exclusively a biological process. So people have not observed methylmercury production by abiotic processes. So we know that the microorganisms have to be there in order to have that production. Now, the transport of that into the food web ultimately leads to both bioaccumulation and biomagnification. And I think this is something that I didn't have a very clear handle on what the difference is between those two. So I wanted to just quickly go through that. So bioaccumulation means that when you have a population of an organism like a fish, that over time, as the organism gets older, more and more of the chemical is being stored in the organism's body, usually in the fat. Like that's usually where these chemicals that we worry about, and specifically methylmercury here, accumulate in the bodies of the organisms. And that's tracking like one type of organism. Biomagnification refers to what happens when you transfer from one level of the food web to another. So here we're showing zooplankton at the bottom, and then fish, and then seals, and then polar bears, which we don't have in Madison, but like because mercury is an issue, even up in the Arctic, people think about that quite a bit also. And so we get both bioaccumulation and biomagnification happening in Lake Mendota, but it only happens as methylmercury. And so when you want to understand where the methylmercury is coming from, how much is entering the food web, and potentially how we can intervene to prevent it from getting into the food web, we have to understand its production. Now, chemists at UW are also trying to understand the processes by which it gets broken down, because, you know, if we could take it from methylmercury and convert it into an inorganic form, like over here, where it gets converted back again, that would be great too. But my research group has chosen to focus on, you know, how it gets produced and where and when. And you'll notice down here that I have this red box around what we call anoxic waters and also sediments. So anoxic means there's no oxygen. And maybe some of you know that Lake Mendota gets depleted in oxygen in the summertime. And that's an issue for fish habitat, as well as the lake just, you know, being stinky if that water, when water comes to the surface and it mixes in the fall. But it turns out that that anoxic conditions is also really serious in terms of methylmercury production. Now, I mentioned already, but I think it's worth reiterating that as a scientist trying to dissect the mercury cycle, we have to think about different aspects of that production. So, you know, you might ask who produces it, what kinds of bacteria, when do they produce it? Because maybe the bacteria are present, but they may not be producing the methylmercury. We don't actually know why they produce it. That is still a mystery in the field of studying mercury methylation. And so there might be some conditions where they're present, but they're not making it. We don't know how much each one can make and how fast they can make it. So questions about the rate at which methylmercury is produced. And then you can ask, well, then what conditions are going to favor its production versus not? Like, are years when we have a lot of, you know, blue-green algae blooms, years that we get more methylmercury production, or years where it's dry, or years where it's wet? And so kind of trying to make more predictive understanding of when it's being produced. And then how easily does that production translate into more methylmercury in the food web? And you kind of have to start by asking just where is it, right? And it turns out that methylmercury is really hard to measure. I think when I entered into this field, I thought that it was just like measuring phosphorus or measuring anything else, but it is really, really hard. And there's only a few research labs in the country or even in the world that can do it and do it well. And we're fortunate to have the U.S. Geological Survey Mercury Lab here in Madison, and they are one of the world experts in doing these measurements. So we partnered with them to do some of those measurements. And so I'll talk about that too. Now, the other thing that I noticed when I first started reading about mercury methylation is that most of the research has been done on the microbes that live in wetlands. So if you open up a textbook and read about mercury methylation, it says that mercury is methylated where there's a lot of, you know, decomposing organic matter. So like dead plants or dead leaves and things like that. And, you know, that often happens in wetlands, and wetlands have a specific kind of chemistry in the water that seems to cause these organisms to proliferate. And so, you know, you think about Cherokee Marsh as being an area where if mercury was being methylated a lot, that it would be going straight into Lake Mendota. And if you found methylmercury in Lake Mendota, that would be the source, would be from the marsh, right? Well, there was some preliminary work done by one of Jim Hurley's students. He never published it, but he started to try to get a sense of where the mercury was coming from in Lake Mendota. And he did just like a quick survey, not a lot of samples, but that preliminary data suggested that it was not coming from the marsh. It was not being methylated in the marsh. Or I mean, it was probably being methylated there, but much more of it was unaccounted for, right? Like it didn't add up where it was coming from. And so that got us really interested. And so we started looking at the literature and found another group at Wisconsin. The one up in the upper left is from a group, including a Wisconsin DNR researcher up at Trout Lake Station in Vilas County, who is studying mercury methylation in lakes up north. And they were able to detect methylmercury in the water at the bottom of the lake where it was anoxic. And so I can explain this graph over here. This is depth in the water column. So 0 is the water surface. And then on the y-axis here, it's plotting the dissolved oxygen. That's these curves right here. There's 2 time points. So there's a summer and a fall. So the fall is squares and the summer is triangles. And then other way around, the squares are summer and the triangles are fall. And what's most important are these points right here, which show the methylmercury. I guess it's more of a rate in terms of the amount of methylmercury produced per day, which is also, it's like even harder to measure than the mercury itself. Like how much, like tracking it over time is very, very difficult. So these are very precious data. And what they found was, this little hint of a peak in the amount of methylmercury being produced at about, in this case, like 12 to 13 meters below the surface. And then also a little bit down here and in the summer. And they did some other analyses too, also for wetland production, and determined that you can't account for the methylmercury measured here by only production in the wetlands, and that there was significant production in the water. And almost no one had documented that before. I think there's one other paper. And as I'll tell you later, that becomes important because if it's being produced like right kind of at the interface between where it gets anoxic and where there's still oxygen, that's where a lot of zooplankton hang out. The zooplankton don't like to be where the oxygen is gone completely, but they will hang out right above where the oxygen is gone because there's a lot of good nutrients and good bacteria that they want to eat. And so they can dip down into that region where there's a lot of methylmercury. So the potential for methylmercury to get into the food web is much higher if that's where the methylmercury is being produced. And so people got very interested in this as an unrecognized source of the methylmercury. So many people were focused on wetlands and sediments, and we said, hey, this is, we're missing something here. And just to convince you that this could be relevant in Mendota, again, if you haven't seen one of these plots before, I'll walk you through it. On the left, of course, I'm showing an animation, and this is courtesy of Madeline McGee, who's a PhD student in civil engineering and now works at the Wisconsin DNR. She ran a model of the Lake Mendota temperature over 100 years, actually. So this shows, these are kind of a weird time frame of 1950 to 1916, but it just shows an annual cycle of air temperature. And then it's linked to this image of Lake Mendota where you can see the temperature being cold and fairly uniform in fall and then into winter. And you eventually get ice cover. I think there's like a little gray bar that will appear on top as you get your ice cover. And this looks like it must be out of sync. December 15th. Yeah, there's your ice. And this is what should be happening in the summertime. I have a feeling that this is out of sync. So let's just focus on what's happening that's blue right now. So in the summertime, the water starts to warm up on top. And as it warms, it gets less dense. And so it floats on top of the cold water that's on the bottom. And, this is why the bottom waters start to smell so bad, because they're isolated from the atmosphere and from the oxygen. And here is sort of in the peak summer where it gets really warm at the surface. And so another way to look at this is on the right here. So the temperature plot, this is called a heat map. And I'm showing the depth of Lake Mendota with 0 being the surface. And then as you go along the horizontal axis, it's a year. So it's each month. And when it's blue, that means it's cold and there's ice on the lake. When the ice comes off, eventually the water starts to warm, and you get what we call this thermal stratification, where it's warm on top, cold on the bottom. And that is almost like a seal on the bottom of the warm pool of water, because it's a barrier to mixing. The physics of it are such that you can't turn the lake over until the water, the difference between the surface and the bottom, becomes less again in the fall, right? So that's why we have a lake that we say is dimictic. It mixes in the spring and it mixes in the fall. Now, the consequence of that in a lake like Mendota is that you get a lot of oxygen depletion in the bottom of the lake. So this plot shows oxygen, where this is high levels of oxygen, the yellow color, and then the darker color is the depleted oxygen. So sometimes, I mean, a lot of the time, we can't even detect it. So there's definitely no fish down there. There's no zooplankton hanging down here at like 16, 18 meters. They really don't like it. But that creates a really good habitat for these bacteria that methylate the mercury. And so this whole period where there's this dark color, we hypothesized that there was mercury that was being converted from the inorganic form into the methylated organic form. And so we set out to make the measurements to show that. And this is another hard to ingest plot, but it's kind of similar to the one that I showed before for the lake in northern Wisconsin. This is depth and then temperature. And so the blue line is temperature. And you can see that it's warmer, like around 18 to 19 degrees toward the surface of the lake. And then you have this really sharp gradient, we call it the thermocline, where the temperature is changing with depth. And that is that point where there's a really sharp difference between the surface and what we call the hypolimnion, or the bottom. And this is the dissolved oxygen. So pretty much homogeneous at the surface. Over here, like at any point in time, it's pretty homogeneous. And then you get this really steep gradient of oxygen. And the mercury methylating bacteria can live all up and down in here, like from about 12, maybe 13 meters down to the sediment of the lake. So that's all habitat for them. They would be happy to make a living there. But what we found was that there was this bulge in the amount of methylmercury that was present at around 13, 14 meters. And there's different ways that you can measure mercury. And this is another one of the things that's complicated about studying it, is that you can look at the total amount. So the total amount here is this green diamond. And then you can look at just the fraction of the total that is methylated, right? And the methylated is the stuff that you worry about the most. And that's the orange triangles. And then you can take the percentage, which is just, using that number and normalizing to this number, and you get the black circles. And when you see a higher percentage of methylmercury, that's an indication that it's actively being produced. And it's important that there's a bulge here and not like a straight line, because if there was a straight line going down that looks like the diamonds, that would mean that it's coming out of the sediments. So the physics of how the methylmercury would be just diffusing in the water column, if it was being produced in the sediments, it would be highest down by the sediments and then gradually get lower as you come up. So, if you imagine that this was the methylmercury, we would say, oh, it's just coming out of the sediments. So it's just like all the other bacteria that people have studied in wetlands and sediments, and it's operating just like what the textbook says it should be. But because we see this shape, that there's no way that it could be coming out of the sediment based on the shape of that curve. And so it's being produced right below the interface of that thermocline, so right where the oxygen starts to go to zero. And so the current hypothesis that we have is that when algae and cyanobacteria, so blue-green algae, are blooming in the lake and die, or if like zooplankton eat them and then the zooplankton die, they settle down through the water column and they actually get trapped at that density gradient, right where the epilimnion, the upper layer, stops and the hypolimnion starts. And that's due to density differences. And so if I was going to plot just sort of like the particle abundance, you would see a peak right here because all that stuff just sits there for a while and decomposes. And so what we think is happening is that those blooms are delivering food for the bacteria that do the methylation. And so you kind of get the double whammy of the oxygen depletion making the habitat for the organisms, and then the algae blooms giving them food and a pretty constant supply during the summer, right? So, we all worry about the algae blooms and the phosphorus in the lake that causes it. And, this is just kind of like another reason why, somebody really needs to figure out how to fix that problem, because it's also promoting the mercury methylation. Now, I decided not to go through all the gory DNA-based work that we do to study the microorganisms and just skip to the punchline. So this is just a cartoon of a microbial community. So 4 different microbes here. And what we found is that the bacteria that are in the textbooks are definitely there. That shows this green one. And they can make methylmercury based on what we see in their DNA. But then there's these other organisms there that had not been shown to make methylmercury before. So you don't read about them in the textbooks or in all the articles that people write about methylmercury production. And that some of them are specialized in breaking down particulate organic matter, which is the sinking phytoplankton. And so these bacteria sort of kick off the process by breaking down that dead stuff and then making food for the rest of the bacteria that also make methylmercury. And so now we're trying to figure out, like, okay, is this one making most of the methylmercury, or is this one, or is this one? Can this one actually do it, even though it looks like it can, but like maybe it's missing one part that allows it to do that? And so we're trying to pull that apart and use some pretty fancy DNA sequencing techniques to figure that out. And so that's kind of where we're at right now, is knowing that there's some interaction between the members of this microbial community, but we don't understand the nature of that interaction and its implications for mercury methylation. So we have some hypotheses, but we have to do the research to figure it out. And I mentioned earlier that this is really important because if you have zooplankton that are moving up and down in the aerobic part of the water, right? That they're aerobes, they need to breathe just like we do. They move up and down during the daily cycle. So at night, often they go, or at night, I think at night they go down to hide from the fish and hang out kind of like right where the oxygen is starting to get low. And that's where most of the methylmercury is, because those bacteria are hanging out there eating all the dead stuff. So it's like this vicious loop. And so those would be the first step going into the fish, which then go into the bigger fish, which go into the ones that we eat for fish fry. So the implications for this that we've just started thinking about is this big blind spot that we didn't know that so much methylmercury was being produced in Lake Mendota, let alone in the water, rather than in Cherokee Marsh or in the sediment. And that this has important ramifications for getting the methylmercury into the food web by zooplankton eating particles that have the methylmercury attached to it. And we really have to go back and revise our textbooks in terms of which microorganisms are doing it and when they do it and why they do it, which are all the things that we're trying to figure out in our research. And yeah, that's exactly why I do what I do, is because it's really fun to have to go back and revise the textbook. So with that, I want to make a few acknowledgements, especially for Ben Peterson, who's the graduate student in my lab who did all of this work. He's like tremendously talented, going off to do a postdoc soon at UC Davis, and is hoping to be a professor someday. So maybe we can hire him back here. And the group at the USGS who runs the Mercury Lab have been amazing collaborators. So if you ever have the chance to run into one of them, you'll be pretty impressed. And then we have funding from various sources, both local and federal. So I think with that, I would be happy to take questions. And I think, should I stop my share while we do questions? Probably yes, right?
We might want to see it if we have to click back to
Okay, sure.
graphics. So let's just get that
That's
back up because we do have some questions for you.
Okay.
But first off, again, Trina, if we were all together next month, you'd be getting a nice round of applause now. You know,
Yeah, I know. Yeah.
But getting to the questions now and our virtual applause in our heads. Let me just pull up the Q&A bracket. We have a good amount of questions here. Here we go. Okay, so first question from Janet. Is there a way to measure the mercury in your body? Now, being older, we handled mercury in high school labs. Of course, we all remember, you know, my generation and older remember the mercury thermometers that we had to use. So what about that?
Yeah, and there's people who have mercury fillings, right? That's actually a pretty significant source of mercury in wastewater, is that people's fillings, you know, can shed a little bit of mercury, or if they just straight up fall out, you know, they go into the sewer. And so there's a lot of mercury in our wastewater because of that. You know, that mercury is inorganic, so it's not accumulating in our bodies. It has to get methylated by the bacteria before it can get in our bodies. But if you're ingesting it in fish, you can measure it in your hair. And that process is actually not that hard. Like, there are a lot of labs that can measure the amounts that are in hair. And the student who did the preliminary work on Lake Mendota actually did this experiment with some friends where they shaved their heads and, well, they measured it, and then they shaved their heads, and then some of them ate fish and some of them didn't. And then when their hair started to grow back, they were monitoring the mercury in their hair, and they could see that if they stopped eating fish, they didn't have mercury in their hair, and the ones that kept eating it did. And I mean, these are like Wisconsin dudes who eat a lot of fish, so, you know, they probably eat more than you and I do even. But yeah, you can absolutely measure it in your hair. I think in adults who are older, it's not nearly as much of a concern, because once all your development is done, then it doesn't have as much of an impact. You have to get a lot to be poisoned if you're an adult. But so it's really women who might be pregnant that we're really worried about, or kids.
Interesting. Yeah, so it sounds like more concerned about what we're actually consuming to get that transfer from mercury versus just environmental factors growing up. Yeah. Okay. Tide is saying, are there any ways to reduce mercury from the lakes and the food web using your understanding of the role that microbes play? Or is it better to focus on improving water quality? Both big questions, of course, with clean lakes.
Yeah, that's a great question. And I don't know the answer. I think, you know, that's part of where our end goal is to answer that, of course. I mean, there's not any treatment method to remove mercury. It's actually present at very small quantities, right? It's like microgram to nanogram quantities. So that's part per billion, part per trillion. But it doesn't take much to bioaccumulate, right? So like, even though it's very low concentrations, it still can get into the fish with, you know, and concentrate. But because it is at such low concentrations in the water, it's very hard to remove. And it would be completely economically impossible to like put all the water through a giant filter or something, right? And so I think focusing on the water quality, right, you're going to get all that benefit of fewer algae blooms, but then also less mercury methylation. So that's probably where we should focus our effort.
Interesting. Another attendee here, anonymous, says, are the impacts of climate change, they talk about the shorter winters, less ice cover, increased temps, have those had an effect on mercury production and availability?
Yeah, we don't know because we haven't been measuring it in our lakes for long enough. And I would say there's not really any ecosystem locally that people have been doing that in. You know, but you can do the thought experiment of the effects of climate change on phosphorus inputs. We can see that if we have a really big rain event in March and April when there's manure on the farm fields and it ends up going into the lake, that we then have worse algae blooms in the summer. And so that, just by what we have shown already, that would cause more mercury methylation if you have more of that dead stuff to keep it going in the summertime. I don't think we have enough data yet to show quantitatively that relationship. You need to watch an ecosystem for a long time to see climate change effects, just because there's so much stochasticity year to year. I guess one fun fact is that at a more global scale, or maybe national scale, like the United States or North America, there's a pretty clear trend downward in the amount of mercury that is coming from the atmosphere. And that's really good news. I think it's partly because of all the progress we've made with acid rain. So regulations about emissions for coal-fired power plants, by regulating the sulfur, we've also taken out the mercury. Also, you can see a pretty sharp downward trend in about 2008, which the mercury researchers attribute that to the economic slowdown that happened in 2008, that there was just less fossil fuel being consumed then. And I don't know if they've actually looked to see if it came back up as economic activity improved, but yeah, I guess the bottom line is that it's going down. And so overall, the amount of mercury going into the ecosystems is less, and that's good news.
Good correlation there. Paul asking, would introducing oxygen where there is none cause a positive or negative, one second, the question drops away. Would introducing oxygen cause a negative or positive reaction in the water? That's the question.
Yeah, that's a great idea. I mean, some people have done that with lakes, usually smaller lakes, putting oxygen in the hypolimnion, and that also can help with the algae blooms, because then you get less phosphorus solubilizing and coming out of the sediment and fueling those algae blooms. Lake Mendota is probably too big to do that. I've actually thought about it because there's some clever engineering techniques that we could use that supposedly would be, not out of reach. Other researchers have looked at adding nitrate, which, at the face of it is seems like a terrible idea because that's adding nutrients to the lake. But the kinds of bacteria that grow, that do the methylation under anaerobic conditions, are inhibited by nitrate. So you could inject a bunch of nitrate into the hypolimnion, and that would inhibit methylation, but you'd risk fertilizing the lake even more and getting more algae blooms. So yeah, we've seen a few reports of researchers trying that, and we're a little skeptical whether that would work here.
Interesting question though, right?
Yeah.
Well, the next question from an anonymous attendee asks, are you aware of any public health studies that look into how mercury affects vulnerable populations or those communities that subsist on freshwater fish in the Yahara watershed? So kind of talking about the different types of people consuming.
Yeah, that one paper that I put up, Stevens was the graduate student that did the work. Last name was Stevens, and then the faculty member who was leading is Pete McIntyre. It's from 2017. So I think I'm going to give my presentation so that people have a copy of it. You can look it up. That's the only one I know of, and I don't know of any follow-up studies. I think it was kind of a one-off thing. We would love to look at that, but I'm not a social science public health person because I'm a microbiologist, and so I'd have to partner with someone, and probably means writing a grant to get the funding to do it. So no, we don't know. And I mean, it would be also interesting to monitor the mercury levels by measuring it in their hair, and making that connection between how much they eat and how much is accumulating in their bodies. But no, we don't know.
And that brings up a good point about the relationship between science and social science, and how as much forward progress there can be made with the physical science or the theory of it. If you don't have that social component, it's a little less tangible. So it's interesting how those two things can connect. So another question is from Richard. He's asking, in the bottom of Mendota, with a high footprint of total mercury concentrations in September, but relatively low concentrations of ME mercury, Me-HG, that one. What is the form of mercury that makes up the difference in concentration?
Oh yeah, great question. So there is the inorganic forms. So like we say it would be, it's more oxidized. So the Hg2 is inorganic, and also elemental mercury, of course, which is more reduced, would be part of that. It also just gets stuck to organic matter. So one of the reasons why it's so hard to study is that mercury in the inorganic form binds really strongly to sulfur. And one piece of interesting fact that I left out is that Mendota has a really high amount of sulfur in it. It has sulfate in the upper water column because just geologically we have a lot of sulfate, and that also fuels the bacteria that do the methylation. And when you get anaerobic conditions or anoxic conditions, so there's no oxygen present, there's also all this sulfur reduction happening, and the reduced sulfur binds the inorganic mercury really strongly. And so in a sense, that's keeping it away from the bacteria. But if it gets really complicated, because if the sulfur organic molecule is small enough, then the bacteria can take it up into their cell and methylate it. But if it's bigger, then the bacteria can't. So the difference between the total and the fraction that's methylated is that material. And there's people that spend their whole career studying how bioavailable the mercury is when it's bound to that sulfur organic molecules. So that's more chemistry. And so our collaborators study that, but that's not something we've got really into. But the sulfur in Lake Mendota is a really big factor in terms of it making more probably than some other lakes.
So we have 8 minutes left, and we have 1, 2, 3, 4, 5, 6, 7 questions. Can we do it? I'll double up on this from Chuck. He asked 2 questions. What are the consequences of overconsumption of mercury in humans? Which is interesting because you were saying not too big of an impact if you're a full-grown adult, but also different question. What is the effect of zebra mussels on the methyl mercury production? So first, overconsumption in humans, and then zebra mussels effects.
Right. Yeah, everything that people have shown that I'm aware of is that once you get past a childbearing age, it would be really hard to eat so much fish that you would get poisoning as an adult. It really is about the development of a fetus and children because of the mechanism that mercury causes toxicity in the body. I mean, if you ate fish like 3 times a day, that, the highest levels, that would probably approach being actually toxic to an adult. But I don't think anybody does that around here. In terms of the, shoot, I forgot the second question, but it was really good. What was it?
Zebra mussels.
Oh, zebra mussels, yeah.
Methyl mercury production.
Yeah, we don't know. They haven't been in the lake long enough, and other people haven't studied. I don't think anybody else has studied that, like in the Great Lakes where there's a lot of zebra mussels. Again, you can do the thought experiment that if the zebra mussels change how the phytoplankton bloom, which is what they've seen in Lake Michigan, then you might predict that there would be less mercury methylation happening because there would be less phytoplankton. Now, Mendota is different though, because it doesn't have zebra mussel habitat everywhere in the lake. It's really just around the edges because of the mucky bottom. They don't like to be on the mucky bottom. And so I think we haven't figured out yet whether that's going to have an impact on the phytoplankton the way it did in Lake Michigan, which is to clear the water. What it's doing is keeping the nutrient cycling kind of at the edges of the lake, which seems to be making more plant weedy stuff grow along the edges, and may also may take some of the nutrients away from the kind of open water area. So we're interested in that, but I think that's also an example of where we have to watch the lake a little longer to see what the effect is.
Okay, Katie asking, how would lake manipulation through mixing, such as the Lake Mixing Project on Crystal Lake in Wisconsin, impact methyl mercury production? Would oxygen levels stay above levels to produce?
That's also a great question. I love the Crystal Lake project. I think, if you could get enough energy into the lake to mix it, then yes, because you would disrupt that interface between the epilimnion and the hypolimnion. And so then you would get oxygen transfer. I think that's a big if though, because Lake Mendota is so much bigger and the stratification is much stronger. So, we've thought about doing that in Mendota, at least my engineering collaborators, but I think it's kind of out of reach in the same way, maybe not quite as out of reach as treating all the water, but yeah, I think that would be hard. That the idea that I had of, it's called a species cone, which delivers oxygen, would also, promote mixing of the water column. So like, that's, I think, the only thing that would be financially or economically viable. I would love to get a grant to try it.
We're loving these questions too. Lots of good ones. Bunny and Ross asking, in tidal water bodies where the water is stirred up, is there less mercury produced or more?
Definitely less, because it gets oxygenated as the water moves in and out. And where would that be relevant? I mean, I don't think there's a lot of mercury methylation in marine coastal systems, but like Lake Erie is so productive that there might be some, but again, it's like more out in the middle of the lake. Yeah.
Going back to, it's kind of social science related, but it's more specific to what you were talking about. Les asking, it seems that cultural factors may exist in Hmong populations leading to high consumption of white bass. African Americans consume more bluegill and have less accumulated mercury compounds. So these are statements. Is there effort to focus on the Hmong community to make them more aware of the dangers of the white bass consumption?
Yeah, the rest of that publication that I mentioned does get into where the different populations are getting their information in terms of consumption advisories and routes to reach those populations more effectively, right? Because there's the DNR website, there's signs, but what they recommend in the paper, I believe, is a more almost individualized effort where people, someone is going to see the fishermen, in person and talking to them about it. We've had some ideas about going to cultural events and, partnering with someone who is Hmong, who can be that bridge to build the trust that's needed and, facilitate that communication. And also trying to have it be a two-way conversation to, hear them and why, more about why they eat particular groups of fish. And so we had that built into one of our grants, but partly because of COVID and like all the things, we haven't gotten to that yet. But again, since I'm not a social scientist, I think if we were really going to take it on, I'd want to work with someone who knows more about how to do that effectively.
All right, well, the broadcast here is going to end in a minute automatically. So we have one additional question, but we'll have to email that to you from Colin. So folks from Clean Lake, you can forward that along. I want to thank Dr. McMahon for your interesting, very, you were able to break down a very complicated topic to folks that don't know a lot about it, like me as well. So it's been very informative. And of course, if you have additional questions, info@cleanlakesalliance.org is where you can send those. We'll forward those to Trina, Dr. Trina. And be sure to circle Wednesday, April 13th on your calendar. That's the next Science Cafe 101, the next Clean Lakes 101 Science Cafe. And that is going to be in person at the Edgewater. We will also have a virtual component if you're more comfortable doing it this way, but hopefully we'll have more people joining for that. And keep your eye on the inbox or on Clean Lakes social media for how to sign up for that next talk. No topic quite yet, but you know, the excitement is it will be in person. So I know everyone wants to turn out for that. So Colin, sorry we didn't get to your question. I just ran out of time here, but we'll make sure to forward that along. And again, Dr. Trina McMahon, very interesting information for something that I'm sure none of us really thought about that much. So real cool. Again, I'm meteorologist Max Saparis. I appreciate you joining us here for the Clean Lakes 101 Science Cafe. And I will see you next month live in person at the Edgewater. We'll talk to you then. Have a good one.
Thank you, everyone.